Yes, we can, Chetan.
Okay. So, a very pleasant good evening to respected speakers, honorable principal sir, respected faculty members, and my dear friends. I, Chetan Sharma, on the behalf of the Department of Biochemistry, Deshbandhu College, bask in welcoming you all in the colorful evening of Holi to the international webinar lecture of our annual academic fest, Biospark 2021, in association with IQAC, Deshbandhu College, under the aegis of DPT Star College Scheme. Before I proceed further, I would like to thank our revered speakers, Dr. Vinod Skaria and Dr. Swati Kunduri, to take out their time and to apprise us of their contribution in tackling rare diseases, which sadly have been rarely discussed about, despite of the excruciating pain and emotional trauma they bring to the survivors. I would like to thank our respected principal, sir, Dr. Rajiv Agarwal, for sharing his crucial time with us and for giving us his consent, and our esteemed faculty members for their constant support and guidance. The world is too complicated in all parts and interconnections to be due to chance alone. I am convinced that the existence of life, with all its order in each of its organisms, is simply too well put together. Each part of a living thing depends on all its other parts to function. How does each part know? How is each part specified at conception? The more one learns of biochemistry, the more unbelievable it becomes, unless there is some type of organizing principle, and this learning is literally incurable for us, the biochemists. And this constant craving for learning was quenched by revolutionizing the idea of the annual fest Biospark by our all time mentor, Dr. Meera Kushal. The Department of Biochemistry, established in the year 1988, has been successful in producing notable alumni like Professor Alo Nag, Dr. Baran Chandra, Dr. Kirti Sharma, Dr. Mahavir Singh, Dr. Garima Gupta, among many others. And we really feel grateful to be a part of this department, which is now one of the science departments being funded by the DBT Star College scheme for research intensive activities at the undergraduate level. Every year, Biospark is inaugurated by the lectures given by eminent scientists working in India and offshores. Last year, in the month of October and November, we conducted an international webinar lecture series on the very relevant areas of health sciences, as well as a hands on workshop on scientific writing, and received a very good participation and response. Biospark lectures are generally followed by student oriented activities like panel discussion, poster presentation, quiz competition, rangoli, etc. This year, we invite student participation in the inter-college competitions in virtual mode like Innovative Idea Presentation termed InnoSpark 21 and Scientific Cartoon Making termed Cell Dialogue 2. Please go through our department website for more details. Taking the legacy forward, here we are with the inaugural lectures of Biospark 2021. And without wasting much of your time, I would like to invite the co-convener for today's international webinar, Dr. Vinita Kashyap, to throw some more light on the theme of the event. Over to you, ma'am. Ma'am, please unmute yourself.
Respected Principal Sir, distinguished speakers, faculty members, and dear students. I, Dr. Vinita Kashyap, on behalf of Department of Biochemistry, Deshbandhu College and IQAC, under the aegis of DBT Star College Scheme, welcomes you all to the international webinar on rare diseases, rare insights into research challenges and concerns. Rare diseases are defined as rare if it affects less than 1 in 2,500 individuals. There are around 7,000 types of rare diseases globally, and about 450 of them have been reported in India. In fact, you all would be surprised to know that rare diseases impact a greater number of people than AIDS and cancer combined. But due to the lack of proper definition, and more importantly, due to the lack of diagnostic infrastructure and systematic data collective system, we do not have an accurate statistics on the prevalence of rare diseases. to apprise the students with various challenges and concerns pertaining to rare diseases like lysosomal storage disorders, cystic fibrosis, CHD, alkaptonuria, autism, to name a few. We have among us two eminent and dedicated scientists who have been pioneering the applications of genomics to diagnose and solve rare genetic diseases in India. I feel honored to welcome Dr. Vinod Zakaria, a senior scientist in CSIR IGIB New Delhi, India, who also co-founded a national network guardian of clinicians and researchers working on rare disease genomics. Thank you, sir, for sparing your precious time. I wish to extend my deep appreciation to Dr. Swati Kunduri, Medical Science Liaison from KC Global Rare Disease from Ohio, USA. She is also a co-founder of an organization concurrent with Canon, which supports the research for medical advancements for children born with rare congenital heart disease. So before I request the convener, Dr. Preeti Karwal, to formally inaugurate the program, let us start this session with the blessings of Lord Ganesha. With this, I would like to invite Akriti, a third year student of biochemistry, to present the Ganesh Vandana, asking for Almighty to make this event a success. So I would like just 2 minutes. Let me
Prabha, you need to share the link again. The sound is not there. Please close this link and share the link.
Ma'am, how to this link sharing is? I'm just sharing the screen actually.
No, don't share the screen. Share the YouTube link.
Okay. Actually, ma'am, I'm not able to find like Chrome tab or something. How to share?
Dr. Preeti, I think we can move ahead if
Okay.
there is some
Okay,
kind
sir.
of.
So now I welcome Dr. Preeti Karwal, the convener of the webinar, to formally inaugurate the program. Over to you, ma'am.
Thank you, Vinita. Thank you so much. Yeah. So on this auspicious occasion of the Festival of Colors and inaugural lecture for Biospark 21, I welcome our esteemed speakers, Dr. Vinod Skaria, Dr. Swati Kunduri, Principal Sir, Dr. Rajiv Agarwal, Dr. Aditya Saxena, IQSC Coordinator, Dr. I.K. Singh, Star College Scheme Coordinator, Dr. Meera Kushal, senior colleagues of my department, Dr. Kamna, Dr. Rajni, and faculty members from Deshbandhu College as well as from other institutions. I extend a very, very warm welcome to all of you and wish you a very, very happy Holi. Exactly after a month of rare diseases, which is on 28th February every year, we are here today to gain rare insights into the challenges and concerns for rare diseases. Now, these concerns are not only faced by few people, they are faced by those 300 million people who suffer from the rare or orphan diseases. And why they're called as orphan? Because there is no pharmaceutical company which is interested in making drugs for these kind of diseases. A disease may be rare, but hope should not be. We have among us our rare disease warriors, Dr. Vinod Skaria, who is a committed scientist to the diagnosis and treatment of rare diseases through his research in precision medicine and prognosis of training and support to clinicians, and Dr. Swati Kunduri, who is committed to improving patient care and outcomes through her close collaborations with healthcare providers and patient advocacy programs. I congratulate you both for your sincere efforts in bringing this rare disease awareness, and also we'll like to know more about the rare diseases from you people. It is my pleasure to welcome our principal, sir, Dr. Rajiv Agrawal, an institutional head for one of the oldest colleges of their university, yet very mature and insightful by experience, who is young at age, but very insightful and mature by experience. He's calm and serene by nature, yet highly dynamic in action. An accomplished academician by profession and mathematician by education, yet an astrophysicist by passion. A hardcore researcher with over 50 international publications in celestial mechanics and space dynamics, having supervised over 10 doctoral and dissertation theses, yet an administrator par excellence, who has launched the institutional growth at an unimaginable trajectory within a very short period of time. A determined leader with a clarity of vision, yet so humble and level-headed. The list of your accomplishments is long, sir. The time is short. So I can't thank you enough for being so encouraging and supporting for our department. And I would now like you to say a few words. Thank you so much for joining us.
Thank you, Dr. Preeti, for the kind introduction. But I have supervised five PhDs and 1 MPhil. So you have to rectify that thing. Thank you again. Thank you, Dr. Preeti.
More in progress. So I counted those also.
Achha, you have counted those. Okay, all right. Then it's all right. Okay. Dear friends and students, a very good afternoon, namaskar, and of course, a very colorful, happy Holi and warm welcome to you all. All of you who are here this afternoon from all around the globe. It gives me great privilege, honor, and happiness to welcome you all to this webinar on the topic Rare Diseases, Rare Insights into the Research, Challenges and Concerns, which is organized by Department of Biochemistry, Deshpande College. I would like to extend my welcome note to our esteemed speakers today, Dr. Vinod Sakeria, Senior Scientist, CSIR, IGIB, and Dr. Swati Kundri, Medical Science Liaison, Ohio, US, and all the participants on behalf of my institution, Deshpande College, University of Delhi. Friends, as I remember that the Department of Biochemistry organized one of the international lecture series in Breast Cancer Awareness Month last year. I want to congratulate Biochemistry Department of our college for taking another initiative for organizing an international webinar on a very challenging public health issue, rare diseases. Friends, a rare disease, as explained by Dr. Preeti, occurs infrequently in a population and is often goes undiagnosed for years because the concerned physicians have never seen the conditions before or use hit and trial methods for the treatment before they land on the accurate diagnosis. Once accurately diagnosed, another phase of battle begins because the physicians may have little evidence or experience to treat them, particularly when a condition is extremely rare and the family of the patient struggles and frequently travels long distances to consult with the few experts who have experience in treating and studying their rare diseases. Because of those problems associated with the rare diseases, the condition is generally emotionally and of course financially devastating for the affected individuals and their families. I'm sure that our esteemed speakers will throw lights on these issues as well. I again welcome our speakers, Dr. Vinod and Dr. Swati, who have unparalleled contribution in raising awareness and improving patient care for their diseases. In the end, I would like to express my gratitude to all the organizers of this webinar, my friends and colleagues at the Department of Biochemistry of my college, Deshpande College, convener of this webinar, Dr. Preeti Karwal, co-conveners, Dr. Vinita Kashyap and Dr. Vanshika Lam, our DBT Star College Scheme Coordinator, Dr. Indrakant Singh, our IQAC Coordinator, Dr. Aditya, to organize this event. I wish all the success for this event. Thank you very much. Dr. Preeti, I think you are mute, I guess.
Yeah, thank you so much, sir. Thank you so much for joining us. Please be with us. Sure. I would now like to invite Kamna ma'am to please introduce our speaker, Dr. Vinod Sakeria.
Yeah. Thanks, Preeti. Am I audible?
Yes, ma'am. Yes, ma'am.
Yeah. So good evening and very warm welcome to you all for today's international webinar on rare diseases, rare insights into the research challenges and concerns. It's a matter of great pride and pleasure for me to welcome our guest speakers, Dr. Vinod Skaria, MBBS PhD, Senior Scientist, CSIR, IGIB, New Delhi, Dr. Swati Kunduri, PhD, Medical Sciences, Liaison, Chiesi, Global Rare Disease Incorporation, Ohio, USA. Our principal, Dr. Rajiv Agarwal, Dr. Indrakant Singh, Coordinator, DBT STAR Scheme, Dr. Aditi Saxena, IQAC Convener, Dr. Preeti Karwal, Convener, Dr. Vinita Kashyap, Co-Convener, Dr. Vanshika Lum, Organizing Secretary, Dr. Rajni Jain, Dr. Prabha Arya, Dr. Asta Giri, Members Organizing Committee for today's webinar, all my distinguished colleagues, including Monica, and worthy students. Congratulations and big thanks to the team of Rajiv sir, Dr. Indrakant and Dr. Aditi Saxena for giving new dimensions, new shape, great height, and recognition to our college. We all know behind every success, there is a teamwork. So thanks to all the team workers who worked off screen. On behalf of the Department of Biochemistry, on behalf of the Department of Biochemistry, it's an honor for me to extend my heartiest welcome, compliments, and felicitations to our distinguished speaker, Dr. Vinod Skaria. Dr. Skaria, Tanzania-born scientist, is a scholar with many decorations, a keen researcher, a crusader for excellence. Dr. Skaria completed his undergraduate studies in medicine from Calicut Medical College with gold medals in biochemistry and physiology. He did PhD in computational biology from University of Pune and worked as a consultant at the Pain Palliative Care Society in Kozhikode. Later, he joined CSIR, IGIB, where he started his career as researcher. He's basically a clinician turned computational biologist. He's involved in creating novel methods and resources for analysis and annotation of genomes and understanding the functional impact of genomic variations. He's also the co-founder of the Genomics for Understanding Rare Diseases, Indian Alliance Network, commonly known as Guardian, which is one of the largest network of 100 clinicians and researchers and 25 medical research centers in India working on rare genetic diseases. He has adopted novel and creative strategies such as use of social media and the participation of large number of undergraduate students in collaborative projects to accelerate genome annotation. Dr. Skaria is best known for sequencing the first Indian genome. He was also instrumental at the sequencing of the first Malaysian genome and first Sri Lankan genome as well. He has also been involved in popularizing personal genomics through meragenome.com and OpenPGX. He was conferred the CSIR Young Scientist Award by Prime Minister of India in September 2012 for biological sciences for developing computational tools to analyze genomic data. He is also an adjunct faculty at Indraprastha Institute of Information Technology. Dr. Skaria's contributions are many. Few notable ones are, he has published numerous papers in international peer-reviewed journals. Few books are also in his credit. He is also the editorial board member of many reputed international journals. He specializes in genome-scale data for precision medicine with reference to pharmacogenetics. In this context, I would like to mention that Department of Biochemistry, Deshbandhu College, very recently before Corona lockdown, on 16th and 17th January 2020, organized a national conference on pharmacovigilance, advancements in the safe drug development, with the special presence of Dr. Ramesh Goyal, Vice Chancellor of Delhi Pharmaceutical Sciences and Research University, Delhi. During this conference, one of the invited speakers had covered this area, pharmacogenetics, in his lecture. That's why I mentioned it here. Dr. Skaria has been pioneering the application of genomics to diagnose and solve rare genetic diseases in India. Today, Dr. Skaria will be enlightening us on the topic, personal genomes to population, experiences from India. So please, all of you cherish with good moments, cherish the good moments with him. Wishing you all safety and good health. Thank you, sir, for sparing your valuable time and precious time. Great to see you. Over to you, Skaria, sir.
Thank you, madam, for the introduction, and hope my slides are visible.
Yes, they are.
Yeah.
Sir.
Yeah. Good afternoon, everybody, and happy Holi to all of you. I come from the CSI Institute of Genomics and Integrative Biology, which is very close to where your college is. So whenever you have time and you feel free, you could drop by to the institute. Before I start my talk, let me introduce what we work on. So my lab works on a variety of aspects covering what we call as genome informatics. Essentially, that means that we work on the computers trying to look at genomes and understand very pertinent aspects of genomes. A variety of problems or questions are being addressed in the lab. This includes how the genome is organized and how the non-coding RNA component of the genome could cause functional correlates. We also work quite extensively with clinicians as part of a consortium called Guardian, which works extensively on Mendelian rare genetic diseases. We also work with clinicians to understand the underpinnings or genetic underpinnings of pharmacogenomic variants and try to implement these findings back into the clinics. We also extensively collaborate with countrywide programs across the world in trying to understand the genetic epidemiology of Mendelian genetic diseases. A small portion of my lab also works in the area of pathogen genomics, where we work on the genomes of tuberculosis and very recently also on viral epidemics. And of course, you could find more information about what we do on our lab website. Now, the basic premise of what we do and why we do is sort of exemplified in this particular slide. We were also very fortunate to be in the right place in the right time to be doing genomics, because as you see here, that the time point when we started doing genomics was also quite exciting time point where the cost of genome sequencing was dwindling very rapidly. And the end result of this exponential drop in the cost and exponential improvements in the scalability of genome sequencing is sort of exemplified in here in the fact that compared to a whole human genome, which was done over a decade's time by multiple different countries, now individual labs could do whole genome sequences. And the outcome of that was essentially a large number of genomes that are being sequenced across the world. Now, this is a slide way back from 2008, and this sort of exemplifies the admixture of the global populations and the genomes that are available at that point in time. And in 2008, when we started looking at whole human genomes, there were around 6 genomes that are available, which includes the reference genome, the Craig Bender and the Jim Watson genomes, and the Chinese, Japanese, and the Korean genomes, all of them which came from quite homogeneous or less admixed populations. Now, what you see in the middle of this panel is essentially a region which is extensively admixed, and this is Middle East and Central and Southeast Asia, almost encompassing a third of the global human population. And what you didn't find here is essentially a human genome representation. And that's essentially where we started looking at human genomes in trying to understand what is the genetic underpinnings of these populations and how can you use that information to build clinically relevant programs and clinically relevant information.
Dr. Vinod, can you be a bit louder?
Yeah, probably I can talk into the mic.
Yes,
yeah. So in 2008, 2009, we put together the first human genome in the country, the 6th country to do so. And the idea of putting together this one single human genome was to understand how to put together the assembly, how to put together the analysis, or in other words, how to learn the tricks of the trade. And thanks to a fantastic set of collaborators, we were able to put together this Indian genome. And it is not really important to do just one genome. We went ahead and also did a quite large number of genomes from this part of the world, which includes the first Sri Lankan. We also contributed to the analysis of the first Malaysian genome and the first Philippine genomes. We are also partners in the Hugo Pan-Asian Population Genomics Initiative, which aims to understand the genetic landscape of the populations of Asia. And very recently, we have also been fortunate to have collaborated with a lot of countrywide programs in the Middle East, trying to understand the genetic landscape of this region. And today we hold quite some enormous databases of its kind for population genomes in this part of the world, which includes the South Asian genomes and exomes for the South Asian populations, Almina for the Middle Eastern populations, and very recently Indigen for the Indian population. Now, one of the applications of looking at these genomes is to essentially understand rare genetic diseases. Now, rare genetic diseases in many ways cumulatively affect close to around 70 million people in India and close to 300 million people worldwide. Now, it's a big problem because of the fact that many of these patients go through multiple clinician visits, go through multiple rounds of misdiagnosis, and lose considerable amount of time and money in terms of attaining the right diagnosis. In many cases, even the right diagnosis is not achieved. And this is largely because there's a lack of general awareness of these genetic conditions. There are 7,000 odd genetic conditions that we're sort of talking about. And there is therefore a lack of an easily available and affordable system to understand what are the prevalent genetic disorders and offer genetic testing. To understand genetic testing, you need to understand the mutational spectrum, which is unfortunately also not available. But of course, we can always look at India as an opportunity, and the opportunity is that we are a large population, and a large population which encompasses a sixth of the world. And unlike many other countries, our genetic pool is diverse and is stratified into small endogamous populations. And endogamous populations essentially mean that these are communities who marry within the same community. And this is a very common practice across the country, that we are categorized or stratified by small cultural or sectarian groups which essentially marry within the same community. And then therefore, you have a chance of amplifying genetic diseases in small communities with a boundary extending within the boundary of the community. But even more important is that we have a close-knit family structure. And this is very important because unlike the rest of the world, or unlike the European world, India has largely been undisplaced in the past 400 to 500 years, because we have never been through major famines or major world wars, and therefore every individual can actually trace their ancestry by 3 to 4 generations, and practically everybody listening to this particular webinar would trace their grandparents. That is essentially 3 generations of the pedigree. So around 7 years ago, we sort of put together this program, which is called Guardian. And the idea of this program was that we would work with clinicians who see rare and undiagnosed patients or families in their clinics. They could refer these patients and families to a program where we would conduct a whole genome or a whole exome sequencing and be able to analyze and interpret and give back the report. Now, on an average year, we would handle close to around 1,000 odd families. And the idea is that we would be able to return back approximately around 40 to 50% of the reports back to the clinician by doing an exome sequencing or doing a genome sequencing. But the rest of the 50% of the cases would require additional genetic studies, additional functional validations to prove the causality of the variation. And that is essentially the science part of what we do. This might take, in many cases, months, in some cases, years. But the idea is that eventually we should be able to give it back to the clinician as a genetic report, therefore impacting the patients and the family. And today, this global consortium has now encompassed close to around 300 odd clinicians from 70 major clinical centers around the country, and in some cases across into neighboring countries in Middle East, Africa, and Southeast Asia, making it one of the largest of its kind in terms of rare genetic diseases in India and this part of the world. And it's not just about sequencing human genomes and giving back reports. The idea is also to take it to research, to amplify education, to improve the diagnosis, and to, of course, help implement policy. And this is one of the recent papers, and for students who could go back, you could find this paper online, where we have put together our experiences in understanding genomics of rare genetic diseases authored by close to around 280 odd collaborators from 70 odd centers across the country. I'll give a few examples of how this would happen. And this is one such example from very close by Agra. And as students, I'm not sure you would have come across this press note, which came in 2015, where parents have asked permission from the President of India to euthanize 6 of their 8 kids. Now think of the plight of the family who has asked permission to euthanize their own kids. And the reason is that these kids, as you see here from the clockwise direction, have a progressive neurological disorder, which eventually makes them succumb to a vegetative state. Now, nevertheless, these kids did not have a diagnosis, did not have a cure, and therefore, this was therefore referred to us in terms of understanding what are the genetic cause for this condition. Now, we did genomic analysis to sort of suggest that these children had a homozygous frameshift deletion in the MLC1 gene with no estimated frequency in public databases. The only other case for this particular genetic mutation was out of Persia, and this sort of confirms the diagnosis of this condition as megalencephalic leukoencephalopathy with subcortical cyst type 1. Or in other words, an extremely rare genetic condition which was previously not very prevalently found in India. Now, we did an extended pedigree for this family, and what we found was quite remarkable. It is a consanguineous community, and just by drawing an extended pedigree, we could find another family member with a kid with a similar genetic condition who also tested positive for this genetic mutation. An extended community survey was done, and we found another 2 families with the same genetic mutation and the same disease. And the results of the community survey was quite remarkable in the sense that around 28% of the family members or the community members in the small community across Agra and across Rajasthan had this particular genetic mutation in a heterozygous state, or in other words, in a carrier state. Now, what would this mean? Now, this would essentially mean that this disease, otherwise with an extremely low prevalence across global populations, could cause the disease in approximately 1 in around 400 children born out of this community. And that's very evident in here because just by an extended community survey with a few hundred individuals, you could identify additional children with the same genetic condition. Now, how do you sort of sort out this problem? The only way to sort out this problem is to improve education and awareness in the community so that the practice of consanguinity could be minimized. But of course, that is not really possible. So the other possibility is to implement and make available genetic tests so that prospective couples can actually test themselves whether they are carriers for this extremely rare genetic condition, but otherwise prevalent in their community. And that's how in a turnaround time of approximately 3 years, we could actually put this diagnostic today in the market, which is available with Dr. Lal Path Labs and also available in the lab free of cost to patients. Now, this is one such case, and this is another case, which is a mid-age businessman who presented with complaints of intermittent fever and joint pain for the 5 years. It was referred as familial Mediterranean fever, an extremely rare genetic condition in this part of the world. An exome sequence and further Sanger sequencing sort of suggested the genetic mutation and confirmed the diagnosis of familial Mediterranean fever. The genetic mutation was a compound heterozygous mutation in the MVFP gene. Now, you would ask this question, how do such extremely rare genetic diseases occur in this country? And of course, you could go back to the genomic data, do a cluster analysis to sort of suggest that this individual had a mixed ancestry. And as the name suggests for the disease, this disease is extremely prevalent in the Middle Eastern populations or in the Mediterranean basin. And this individual had an ancestry which traced back to the Middle Eastern populations. And this is not just one family. Just to give an example, there are many, many more families, many, many more communities which trace an ancestry from the Middle Eastern population, and then therefore could have genetic components which trace an ancestry from a different part of the world, and therefore diseases which are prevalent in a different part of the world. And that's how we also started collaborating extensively with a large number of countries in the Middle Eastern population, trying to understand what the genetic diseases which are very prevalent in those populations so that we can help those populations back here in India and vice versa. And then comes to a very pertinent question about what is the technology and how is technology moving. And what we have been doing all these years was to use whole exome sequencing, which is essentially to sequence just 2% of the genome, which encompasses protein coding genes and protein coding exons. And what we see here today is essentially to also state that in terms of the time for the analysis, in terms of the cost of sequencing, whole genome sequencing has slowly surpassed the time scale as well as the cost of whole exome sequencing. Or in other words, the future diagnostics of choice for many such genetic conditions would indeed be whole genome sequencing, with the advantage that it could have applications in the clinic, like in neonatal ICUs, because you could offer fast turnaround for genome sequencing, which is already happening in some parts of the world and being piloted in India in our labs. It could identify familiar genetic diseases and of course identify genetic mutations which are outside protein coding genes, which we now increasingly see in many of the families which remain unsolved using whole exome sequencing. And it is, in our opinion, going to be the most innovative diagnostic because it encompasses all genetic tests into one, which encompasses single nucleotide mutations, which includes protein coding gene variants, which includes structural variants, which includes chromosomal abnormalities, and of course, indeed, repeat expansions. Now, as students, what you need to keep in mind is that almost all the definitions of rare genetic conditions are actually wrong. And this is essentially because genetic conditions are identified by clinicians. And for patients to come to the clinician, they need to have a valid reason to come to a clinician. And in many cases, what we also realize is that for diseases or traits which are not deleterious to an individual in terms of the functionality of the individual, many of these conditions remain undiagnosed because they will never ever come to a clinician. And this is one such condition. This is nothing but hypogonadotropic hypogonadism, which is you can have a perfectly normal life except for the fact that you cannot sexually reproduce. And this is such one family referred to as from SMS Medical College, Jaipur, Professor Sandeep Mathur, extensively followed up for the last 5 years. And just to imply that this extensive follow-up of the 5 years was essentially to identify additional individuals in the family because nobody in the family would be forthcoming to come to the clinician and state that you had hypogonadotropic hypogonadism. And this points to the fact that the rare disease estimates in any country from clinical data is essentially error-prone. So therefore, you need to have mechanisms of identifying genetic diseases in a country and in the community. And the only option to do that is to essentially sequence normal individuals across the country. And that is the idea of sequencing over 1,000 odd individuals as part of the Indigen program. And the idea of this program was to sequence large populations across the country and with the idea of understanding frequencies of pathogenic genetic mutations in the community to identify founder effects and of course understand pharmacogenomic landscape of the population. And much more importantly, to use this information and data to create commercially viable products so that they can be rolled out across the country. And as part of this program, we also have a card and an app which are provided to each of these participants, over 1,000 odd individuals. And using this card and the app, you can access your genomic information in a secure, privacy protected, scalable, and clinician enabled fashion, which can actually make a lot of difference in your clinical decisions, not just in terms of pharmacogenomics, but also in terms of reproductive decisions by accessing carrier frequencies of diseases, in terms of long-term care, in terms of accessing incidental findings in your genome. And one such example is what we sort of put together from this program is the pharmacogenomic landscape of the Indian population. And what we find is that there are around 27 unique variants of clinical actionability in the 1,000-odd individuals whom they are sequenced, and every individual would carry approximately 4 to 6 clinically actionable pharmacogenomic variant in their genome. And many of these have very significant allele frequency differences compared to the global population. And just to suggest that they have different allele frequency distributions compared to the global populations listed out here. So in other words, this sort of forms a template in terms of understanding what are the prevalent pharmacogenomic variants in the population, not just to understand what are these prevalence estimates, but also to implement pharmacogenomics in clinical testing policies, build better diagnostics so that these can be implemented in clinical settings, and so on and so forth. And the other applications we have sort of come out from this program is essentially an extensive genome scale carrier screening application for prospective couples who are interested in understanding whether they're carriers for a particular genetic condition. And this screen today encompasses close to around 1,700 prevalent genetic diseases, 1,300 odd genes, and 10,000 odd genetic variants, which are clinically annotated and put together into an array so that they can be deployed across large numbers of individuals. In summary, before I close, the idea of this entire program was to essentially create an ecosystem for clinical genomics in the country, right from working with clinicians as part of the Guardian Consortium in terms of educating clinicians through multiple education avenues, implementing testing programs as part of the GOMED, which is run by my colleague Dr. Mohammed Farooq, which today provides over 130 odd high-end genetic tests, which are cheap, affordable, and accessible to community members. By running this over, creating evidence, creating tests, and implementing value to the diagnostic industry by licensing these technologies out to commercial diagnostic laboratories so that they can take it further to a more large number of individuals, not just in India, but across this continent. By understanding the frequency of genetic variations through the indigent program in terms of what we call as genetic epidemiology, and of course, by putting this all together into implementing research for discovery of drugs for many of these rare and undiagnosed genetic conditions. I think I'll stop there and take questions, but before that, let me acknowledge a lot of people who made this possible, including a lot of students who are presently in the lab and have moved out, fantastic set of clinical collaborators from across the country and collaborators from across this region, Sridhar Shivasubbu for most of the NGS that was put together, and consistent funding from CSR over a decade to put together this program and clinical implementation. I think I'll stop there and take questions, but before that, let me also put in a slide for interested students. There is a program that we have been running. This is the second edition of this program, a 16-week online course on how to systematically approach genomic variant analysis and clinical interpretation. Thank you very much.
Thank you, sir, for a very delightful and a very insightful talk. I would like Dr. Vanchika Kashyap to take the questions posted in the chat box.
Although no questions have been posted till yet, I would like to ask question to Dr. Skaria. Apart from the diseases you had mentioned, there are some other rare diseases also which are being seen in the children, especially the autism spectrum disorder. And there are any other mutations? There are some mutation. Any idea about the mutation which has been mapped for that spectrum disorder?
Yes, so many disorders. So what I sort of suggested in the slides are not an exhaustive category of all genetic disorders that we handle. And the program is essentially to understand disorders which don't have a diagnosis, right? So if there is a patient who is diagnosed as sickle cell anemia or thalassemia, probably we are not the right people to go about doing that because they already have a diagnosis. If you already have autism spectrum disorder, then probably you already have a diagnosis. In many cases, these are patients who don't have a diagnosis. In other words, even with the best of the clinical scenario, they don't have a molecular diagnosis to prove or suggest what is the disease that they have.
As per my as per my studies, I have not came across any kind of specific diagnostic system for autism spectrum disorder. There is only the question-based diagnosis which occurs through doctor and to the patient. So, what exactly the diagnostic you meant for a clinical diagnostic or uh, question or interactive based
Yeah,
diagnostic?
so not all diseases are genetic, right? A lot of genetic diseases, what we sort of come across or what we sort of say are genetic disorders are not really genetic disorders. The best example would be common disorders like, for example, diabetes and autism, diabetes and hypertension. These are not genetic diseases. These are environmental diseases with a small genetic component, and therefore, this genomics has no role in diagnosis of such conditions. And the same is for a lot of other conditions in pediatric neurology, including autism spectrum disorders. A significant large percentage of the autism spectrum disorders do not have a very definitive genomic marker for diagnosis. And then therefore, genomics has, I would say, not much of a role in confirming the diagnosis of autism spectrum disorders. But having said that, a small subset of patients with autism spectrum disorders, we do have come across that they have metabolic disorders, which get mislabeled clinically as autism spectrum disorders. And in many ways, such a mislabeling or misclassification can actually prevent them from accessing the right kind of therapies or right kind of treatments. The idea of the program is if there's a familial occurrence of such condition, when I say familial, that necessarily means that 2 members in 3 generations, then they could access this program through the referral system, through a clinician, and if they turn out positive for a metabolic disorder, then they could access the right kind of treatment.
Okay. So there's another question. What are the challenges one can face in establishing the lab for diagnosis for genetic disorders?
I don't think there is any challenge for say establishing a lab. The biggest challenge is, of course, getting grants to sort of put together things. And
Dr. Vinod, you're not audible. We cannot hear you.
So you are not audible.
am I audible now?
Yeah.
Yeah. Can you repeat?
What were you saying?
Yeah. What I was trying to say was, apart from the biggest challenge in sort of setting up the lab is to essentially get grants to set up the lab. Apart from that, the technologies are really widely available across. The biggest challenge, of course, is to get grants.
There's another question that are there some measures to identify the symptoms of genetic disorders? They are specifically which are related to the genetic disorders only.
No, there are no symptoms which are specific for genetic disorders. Now, many of these genetic disorders, as I said, encompass a very large number of conditions. There are around 7,000 odd conditions, around 1,500 to 1,700 of disorders which are very prevalent in the population. There is nothing very common between them. The only thing common between them is probably that they would occur in a familial fashion, or they would occur within the same communities, or recurrence in the same community or the same society, and therefore that is sort of a clue to suggest that these are genetic conditions. And other than that, there is nothing very common that in terms of symptoms or in terms of signs.
Okay, sir. So have you worked on epileptic seizures also?
Yeah, so neonatal epilepsy is something of interest to us because many of the metabolic disorders do present as neonatal epilepsy disorders. In many ways, providing a right molecular diagnosis for these patients, they can access the right kind of treatment. And fortunately, many of these conditions are actually now treatable.
Okay. So there is one more question. How much is the cost difference between the whole genome sequencing and whole exome sequencing? And how about the UTR or regulatory regions that may be missed in WES?
Right. In terms of the accurate cost, of course, that would vary between the settings that you access these costs. But what I would say is between whole exome and whole genome today, the cost difference is by a factor of 2, or in other words, whole genomes are today twice expensive as whole exome sequencing. Yes, whole exome sequencing does not in many ways cover the entire UTIs and the entire regulatory regions. That has been a limitation. But having said that, the corpus of information that we know today for genetic diseases, a large number of these variants do not occur in the regulatory and the promoter regions. Now, that is of course a limitation because the way we have traditionally looked at genetic disorders and traditionally the way we have sequenced genes and regions across genes. But having said that, as genome sequencing becomes pervasive, a lot of these new genetic variations which occur in promoter and regulator regions would be uncovered, and that essentially is going to be the future of genetics and genomics.
So one more question. Can copy number variation also be covered in WES?
So large copy number variations, when I say large copy number variations, which I would mean to say that tens of thousands of kilobases of copy number variations can be covered on WS, not very accurately, but quite effectively. But small copy number variations, uh, uh, whole exome sequencing is probably not the right approach. The many alternative approaches that people have used today, this includes microarrays, comparative genomic hybridization, etc., and MLPA for very small chromosomal deletions and duplications. But having said that, that's what I sort of said is uh, today when people are doing sort of a stratified approach where first you do a whole exome sequencing and then do a chromosomal microarray, or vice versa, where you do a chromosomal microarray to exclude duplications and deletions and then to do a whole exome sequencing, of course this incurs a lot of cost and of course loss of a lot of time. And that is probably where whole genome sequencing tomorrow is going to make a difference, because I said already the cost of whole genome sequencing is just a factor of 2 compared to whole exome sequencing, and a triaged approach for a chromosome plus whole exome sequencing can pretty much come to a value close to a whole genome sequencing. But nevertheless, whole genome sequencing can cover big and small chromosomal deletions and duplications or copy number variations.
How much DNA amount is typically required for whole exome sequencing?
Very small amount, I would say something around 10 nanograms would be good enough.
And the DNA would be isolated from the blood cells only. To start with, how much the volume we should take?
So nothing very specific about that. So it depends upon what you want to look for. So blood cells essentially sort of are derived from the mesenchymal cells, and therefore sort of roughly replicates the genetic makeup of an individual. Now, having said that, there can also be other samples, like for example, uh, you could take the skin, right? A lot of skin disorders could have somatic origins for genetic mutations, and then therefore the best opportunity to look at those disorders is to actually look at the skin samples. There could also be other samples, like for example urine, which is very good to look at mitochondrial genetic disorders, or for example muscle biopsies, which are very good at looking at mitochondrial genetic disorders. So depending upon the disorders, depending upon the questions that you're asking, your sample type might vary. But generally speaking, when you plan to look at the genetic mutations which are representative of what you call your heritability, then typically you take blood cells, and of course you in the blood cells you take the WBCs.
This technique can also be followed for prenatal diagnosis.
Yes, whole exome and whole genome sequencing are not really into prenatal diagnostics today, because prenatal diagnostics mostly rely on very specific gene amplification and sequencing, or very specific chromosomal duplication deletion, either to karyotyping or through very specific fish. Now, there is a alternative to this, what is called as non-invasive prenatal screening, or what we call as NIPT. And NIPT essentially relies on the fact that there are the chromosomes of the child in the mother's body, and then therefore from 10 ml of blood from the mother, you can pretty much sequence enough of the child's DNA to understand very specific chromosomal duplications and deletions. So this is a technology which is fast moving, and what we think is in the future, apart from just chromosomal duplications and deletions, you could actually get much higher coverage of genomes to understand very specific genetic mutations which could occur in the child.
Okay. So another question is, like, if someone is tested positive for genetic disorder, how the treatment can be done with the medication? How the medications could be followed for the treatment?
Right. So not all genetic disorders have treatments. That is a fair assumption to make. Approximately, I would say 20% of the prevalent genetic disorders that occur in the population, in an Indian population, or for that matter, in our cohort, could access either a very specific treatment because of the molecular event that has occurred in that family, or a non-specific treatment which can sort of manage the condition. Now, to give some examples, what I could sort of suggest is, like for example, the best cases would be metabolic disorders, like for example, phenylketonuria, where very specific metabolite restrictions or food without a certain set of metabolites can be used to essentially treat such children. And this is not very what you call broadly used, but in many cases this is retrospective. After understanding the molecular defect, we could sort of understand what are the treatments that can be employed for these children. There's no broad guidelines on many of these treatments. Some treatments, yes, some treatments, no. Like for example, other class of disorders is primary immunodeficiency conditions, where a class of immunodeficiency conditions, the children don't make, for example, antibodies. So they could be supplemented from outside by IV immunoglobulins, or they could undergo a hematopoietic stem cell transplant so that they can get in stem cells which can produce B cells and antibodies. So it depends upon the molecular defect that you identify, the treatment decisions are made.
Which technique would be more preferred, WGS or WES for diagnostic for genetic disorders?
Yeah, so if cost was never an issue, whole genome sequencing obviously would be the best choice. But of course, in India and many other countries, cost is of a major concern. And then therefore, we sort of resort to much cheaper and scalable approaches going forward.
So how does one distinguish fetal DNA from maternal DNA for non-invasive cell-free prenatal DNA diagnosis?
So in fact, you don't really differentiate. So depending upon the methodologies, in one of the methodologies, you don't really differentiate. You just sequence the DNA which is available together. But looking at the number of DNA fragments arising from chromosomal regions, if you have, for example, what do you call a chromosomal deletion or a chromosomal duplication, like for example in Down syndrome, then the number of reads that map to that particular chromosomal region would be much smaller for the fetal fraction compared to the maternal fraction. Now, there are also computational ways to differentiate between fetal and maternal fractions depending upon the number of variations which are shared between the pieces of DNA. But these are not physical methods. So essentially what you take is you separate out the plasma, and what the majority of the maternal DNA is essentially in cells in the WBCs, which are not really in the plasma. So therefore, you sort of enrich for the fetal fraction compared to the maternal DNA. So in many ways, you cannot absolutely identify what is fetal and what is maternal, but you have methods which can suggest what is fetal and what is maternal.
Yes, sir. So can you suggest some genetic disorders one can study in the young undergrad level population using a simple invasive sample collection?
Yeah. So yeah, one of the most prevalent, what you call genetic, I will not call it a disorder, but a genetic trait is a genetic trait which affects smell and taste. Because a variety of individuals have genetic polymorphisms in the olfactory receptor genes, which could preclude them from tasting a certain set of molecules. And one of these genetic mutation is in a gene called TASR, and there are essentially molecules which these individuals cannot taste. So this might be a good example. It has no clinical implications whatsoever, but nevertheless a good example to sort of understand prevalence of such mutations in the population. And there is of course a biochemical test where you can actually taste a strip coated with a particular molecule.
We do more PITC testing,
That's right.
tasters versus non-tasters.
Absolutely. So this is because of a genetic mutation.
Apart from that,
what is really prevalent in the population is something like beta thalassemia. The carrier frequencies of people for beta thalassemia is in this part of the world is at least something around 6 per 100, or approximately 6%. So in a class of 100 individuals, you would find a significant number of carriers.
so why copy number variation can't be detected by WES?
Now, whole exome sequencing is essentially depend upon the fact that you sort of capture specific regions in the chromosome using probes. Now, when you capture specific regions in the chromosome using probes, you have sort of lost the information about the ratios because you're sort of enriching for certain regions using an affinity-based purification method. And then therefore, copy number changes are not very sensitive on such an approach. But having said that, if you have a homozygous deletion where you have lost both the copies of the DNA, then of course in a whole exome sequencing, there will be no capture around that region, and therefore you could suggest. But in terms of copy number duplications of, say, ideally you should have 2 copies, but if you're expecting a 3 copy, it may not be very accurate on a whole exome sequencing because of the methodology of capture which is used. In a whole genome sequencing, you don't have a capture methodology, and then therefore you can compare ratios of the reads in the region and outside the region, and these are pretty much more accurate therefore than whole exome sequencing.
So is there any possibility that the genetic disorders can be treated using natural chemicals obtained from plants or animals?
The answer is probably yes and also no. Some genetic disorders could be treated by products which are derived from nature. And in fact, many of these drugs and therapeutics that we sort of think are products of nature, but nevertheless made in pure isoforms using chemical methodologies, but not really isolated from natural products. So yeah, that is true for practically all therapeutics around. Many of the therapeutics that we commonly use are indeed products of nature, but synthesized to purity because of the issues with purity of natural products.
So it is said that the rare diseases are generally, it is not necessary that rare diseases should have a genetic component. So what could be the other way to address those conditions which do not associate with any kind of a genetic component? There are some epigenetic changes in those conditions, or there are other reasons for?
Yes, so rare diseases are not always genetic conditions. I mean, that is very understandable. And therefore, genomics and genetics cannot be used to identify or treat such conditions because for the fact that they are not genetic. Now, rare is by definition of a frequency of occurrence of a particular condition, and therefore there is no underpinning that rare equal to genetics. There's nothing like that. But having said that, there are also genetic conditions, and most of these genetic conditions are rare, so they more oftenly are used in synonym to each other. But having said that, yes, there are epigenetic conditions, there are environmental conditions which are also rare in occurrence, but without a genetic cause.
So it is the question now is specific to the case study you have shown to us, that where a man is having a rare deletion who is suffering from an intermittent fever and a joint pain. So the symptoms are coinciding with the symptoms of arthritis. So how can one be sure for the diagnosis for the rare disease?
Very interesting question. So this individual had gone and got tested for multiple times over 4 to 5 years, right? And that essentially is a problem for many of these genetic conditions, that they get diagnosed as something else, which are much more commoner, right? But the clue to such a diagnosis is the very fact that the individual tests negative consistently over years, right? And then therefore the suspicion that there's something else that is happening, right? On the first look, on the first admission, yes, in many ways these conditions would never be diagnosed. But of course, the clue to such a diagnosis is that the recurrence of the event and recurrent negative reports for a consistent, what they call common disorder that they present with.
Okay, sir. So any of the rare condition is also seen for the behavioral changes in the patients?
What do you mean by behavioral changes?
Behavioral changes mean extreme behavior changes, like not a normal case.
Yeah, so psychiatric abnormalities.
Psychiatric
Yeah, psychiatric
abnormalities.
abnormalities are indeed common in some of the genetic conditions, essentially metabolic disorders, but not in all. Yeah, but there are indeed disorders. One of the best example would be Wilson's disease. Wilson's disease, I'm sure you would have all learned, it's a disease of copper metabolism caused by a mutation in the ATP7B gene, which is a copper transporter. Now, one of the commonest presentation of Wilson's disease is actually neurological symptoms. They could present as mood disorders, they could present as schizophrenia, they could present as frank psychiatric abnormalities. In many ways, they are also treated for the psychiatric abnormalities without being tested for Wilson's disease. So, yes, indeed, some genetic disorders could present as psychiatric disorders.
By how much time the diagnosis is done through WGS? How much time generally it takes?
No timeline. So depends upon the kind of cases that you look for. Like, for example, if you know you have beta thalassemia, you exactly know what gene to sequence, you exactly know what mutation to look for, then the diagnosis would be much faster. In terms of the sequencing time, the sequencing time is not much. From sample to sequencing for a whole genome sequencing is today less than 24 hours. And in fact, you can sequence close to 30 genomes in 24 hours. So time in terms of the sequencing is not much of an issue. Time to the diagnosis is probably much longer because of the fact that you need many more clinical correlates to sort of match and look for specific variations, and then of course more importantly confirm those genetic variations. And confirmation of the genetic variations is important because just finding a genetic mutation in an individual with a genetic disorder doesn't mean anything. You need to prove that these are causative. For that, there are additional assays that you need to do in many ways. So that actually takes a lot of time.
No, there are no more questions. Thank you, sir. Thank you so much for patiently answering our questions. Over to you, ma'am.
Yeah, thank you so much, Dr. Vinod. Thank you so much for such a good talk and inspiring our students for doing this genomic variant analysis and clinical interpretation. And I really want to thank you personally for allowing me and my students to join your lab for some time. And we actually enjoyed that a lot. We learned so many things in that phase that we are still using that. And the students are also still thankful. And some of the students actually got through one of the awards also, the travel award that they got for Hargobind Khurana Award. And their students are also there. They're already there in this webinar group also. I would like to invite Vinita to give a formal vote of thanks to him.
Yeah, thanks a lot, sir. It was a very delightful and an insightful talk. It is very, even on the eve of Holi, you have been, taken out your precious time to give such a detailed talk on the personal genomics. And Thank you very much, sir, for patiently answering all the queries so thank you very much, sir, for sparing your valuable time today. And we really have, our students have really learned a lot from your talk. And definitely, I will, all the students of our department would like to join the online the online training course, which you have suggested. So, sir, if it is possible, you can share the link with us, with either in the chat box or either with Preeti ma'am, so that we can forward it to our students. And now, so thank you very much, sir.
Thank you very much, and it was indeed my pleasure. And yeah, we had a fortunate time working with students and Dr. Preeti, and which has actually resulted in quite a number of students actually learning the concepts of more than the concepts, the real problems in genomics, which actually precludes it from reaching out to the common man. It was indeed a pleasure. Thank you very much.
Yeah, thank you, sir. Thanks a lot, sir. Now I would like to play the, unfortunately, because of some technical glitches, we were not able to play the video by our third year student Akriti. So before we, our next distinguished speaker has already joined us, Dr. Swati Kunduri. So before that, let me play the video, the dance performance on Ganesh Verma.
Better luck next time, Akriti. I think this video is not going to play.
Ma'am, is the voice coming?
No, it's not coming. I think we can leave it. We can play it for some other event, the second event maybe. And Dr. Swati is already there, so we should get started with the talk.
Okay, ma'am, I'll stop sharing it. Sorry. Yeah.
Can you hear me?
Yeah, we can hear you.
Yes, Swati, we can hear you and see you.
Okay,
yeah,
perfect.
Thank you so much. Thank you so much for joining this early morning. Yeah.
My pleasure. Let me know when you can see my screen.
Yeah, let me just introduce you and then you can share your screen. Yeah. So Dr. Swati Kunduri is with us. She's a medical science liaison working in KC Global Rare Disease, Ohio, US. As a medical science liaison, she represents her healthcare company to the decision makers in the medical community, that is clinicians and key opinion leaders. She also began her journey from University of Delhi, like many of us here. She did her bachelor's from Miranda House, followed by master's in biomedical sciences from ACBR, Delhi University, in the year 2006. She then moved to US and worked as a research assistant in oncology at West Virginia University. She did her doctorate from the same university in pharmaceutical and pharmacological sciences in year 2012. While working as a research associate at West Virginia University Medicine, she spearheaded a project on mechanism of action of AD1 receptor mediated smooth muscle contraction. Besides the regular postdoctoral programs that she did, she also did certain certification courses in the design and interpretation of clinical trials at Johns Hopkins University and medical science liaison training. Prior to joining her current role in KC, she also worked with University of Cincinnati as translational research liaison for 3 years, where she collaborated with scientists and physicians at the University of Stanford and Mount Sinai Hospital for a project on life-threatening arrhythmia. She has excellent publications and has won several awards in presentations, in her research presentations. She also volunteers for science communication and career counseling for undergrad students. Besides her job, she has co-founded a non-profit organization in the year 2018 with her husband to support families of kids with neurodevelopmental, congenital heart and rare diseases. And she's supporting them emotionally, financially, and spiritually. She, being a science communicator, actively provides consultation and public speaking for congenital heart defect awareness and improving family-centered care. She organizes and manages a lot of events for patient families and leads several fundraising projects and patient advocacy programs for families with children in ICU at Cincinnati Children's Hospital Medical Center. So Swati, you're already there. Thank you so much for accepting the offer. And you're there early morning, and thank you so much. And I wish you a very, very happy Holi. And please, we want to hear you now.
Thank you,
Dr.
Preeti,
Vinod
for
is
that.
also there with us. He just delivered his talk. So, and we we gave a lot of questions to him, and he kept answering patiently.
Yes.
And we believe it is going to happen for you also.
We'll see. Sure. Thank you. Can you all see my screen?
We cannot see your screen yet.
What can you see?
Hope others have stopped the screen share. Yeah, now it is getting started.
Is that it?
Yeah, we can see it. We can see it.
Thank you, Preeti, for that kind introduction. I am out of Delhi University, so I was really excited when Preeti reached out to me. It's always an honor and privilege to come back to your university and remember all those good days from Miranda House, eating chaat and bhelpuri at Hindu, and just those all fun days and of fests and stuff. So I really thank Preeti for inviting me for this, and Dr. Sakaria. That was a great talk and a lot of learning for me as well. So I will touch base with you on that later, but I'll go ahead and start my talk today, which is essentially based out of what I do personally and professionally, talking and connecting to not only clinicians and providers, but also connecting to patients, as Preeti mentioned, with rare diseases. And I come from another side of what Dr. Sakaria had very eloquently mentioned in his talk. I deal with a lot more of a patient advocacy and looking at the patient's perspective as well as the clinician's perspective, the challenges that they go through through this odyssey that they describe it as. So my title from that is called The Zebra Among the Horses. And before I go on with the talk, I want to put out a quick disclosure that the views and opinions expressed in this presentation are those of my own and do not reflect the standards or practices at any organization that I work with or have been affiliated with. And before this talk, I just wanted to let you know, I'm going to give you a quick talk, but also essentially feel free to interject with me with questions when you don't understand or when you feel like it can be more of a discussion, because I think the Zoom session does allow that. So feel free at any time, but I will leave that to the host, Preeti and her team, how they would like to take about it. But I'm okay with if you want to interrupt me anytime with questions. So when I first came across when Preeti was asking me the topic, I was really thinking, but it essentially emanates from the fact that when medical students in the US are taught how to diagnose illnesses, the students are trained to be on a lookout for common ailments and not look for rare illnesses. When symptoms match more than one illness, the most likely diagnosis is the most common among all the possibilities. That makes logical sense. What is the most likely diagnosis? And it's more likely to be a common horse than a rare zebra. But as you hear over and over from these rare disease patients, that zebras do exist. Physicians are often taught, or young residents in training are often taught, that when you hear hoofbeats, think horses and not zebras. But when you hear a rare disease patient talk, now he says that the zebras do exist and physicians need to be more aware. And this, as I go through the talk, will be echoed both by the physicians and the patients as well, that physicians need to be more aware of when to go beyond the horse to spot that zebra, because the longer they suspect a horse, when it really is a zebra, the longer it will take to get to the correct diagnosis. And that means a longer time without treatment. Currently, there are 7,000 rare diseases, and in the US, it's estimated to be, and I say that as an estimate, to be 25 to 30 million, because there are still a lot of rare diseases that haven't been discovered or patients who haven't been found with those conditions. There is a heterogeneity in the rare disease definition, and I'm sure Dr. Sakaria did allude to it, that the World Health Organization defines rare disease as a disease with prevalence of 1 or less per 1,000. And then the United States, the Orphan Drug Act of 1983 defines rare disease as a condition that affects fewer than 200,000 people in the US. As I mentioned before, 25 to 30 million are affected by it. And in the European Union, the rare disease is defined as a rare when it affects fewer than 1 in 2,000 people. And in India, if it affects, from my last research, that affects fewer than 1 in 2,500 people. And it's called as a rare disease. Despite the heterogeneity in the rare disease definition by different countries and standards, the rare diseases are essentially a spectrum of diseases and disorders with different etiologies. But the most common denominator being the infrequency of their occurrence in populations, with most diseases having no defined diagnostic or treatment protocols available, as even Dr. Sakaria went over. While each rare disease affects a small number of people, together rare diseases affect more than 25 million Americans here. And that is, I wouldn't say a similar number, but there are a lot of rare diseases that affect a lot more larger population, but it's the lack of a proper treatment protocol or diagnostic. More than 90% of the rare diseases have currently no FDA approved treatment. And many of these rare diseases, not all, are genetic disorder, which do have an autosomal recessive or dominant inheritance, and not all have the same prevalence. In my next few slides, I will take you over a survey that was done essentially in 3,500 patients who had 436 rare disease conditions. And these patients, and as I go through the talk, I will talk about picking out a few of those rare diseases to help you understand the prevalence and the rarity in it with different rare diseases that comes through. I know this is a quick snapshot, but I will go through the table in more detail. And I wrote this as this is a diagnostic odyssey of a rare disease patient is essentially what my talk is about. And I say this as an odyssey and not a journey is because when you talk to these patients, they will tell you that please don't call it a journey, because a journey sounds like something you would find enjoyable. And to call it an odyssey because it is more accurate, because it is not a fun experience, but more of a lot of eventful. And like I mentioned, this study that looks at the survey that looks at nearly 3,500 patients and caregivers across 436 different diseases. This table on the right looks, fits in 61 different diseases and talks about the median average for diagnosis was up to 4.4 years. And nearly 46% were misdiagnosed, because a constant theme in the rare disease community is differential diagnosis with what we called as a horse previously, or what is most commonly known in the rare disease in the community, or what there has been more awareness. And nearly 47% believed that their treatment was delayed because of the misdiagnosis. Just zooming into the table a little bit more, if you look at the diseases that are in red, that is like the spondylitis, Fabry disease, as well as the mastocytosis or Pompe disease, a primary immune deficiency. You can see that I don't know how clear it is to you, but you can clearly see that these diseases have a median time for diagnosis of over 10 years. That means when these patients come in, they essentially are not diagnosed for a longer time because there is not so much education in the system for it, or there are not diagnostic tools available for it, that these patients keep on going back and forth. And I will allude more to the patient voices more than mine to tell you more about how these patients essentially sometimes find themselves in a roadblock because there hasn't been enough done for their diagnostic treatments. Or even if it's done, the awareness or education, there are knowledge gaps in the system. And the three diseases that are in the yellow, highlighted in the yellow, like cystic fibrosis, Ehlers-Danlos syndrome, as well as hemophilia, essentially identifies those three different rare diseases which are prevalent and well-known, at the same time another spectrum where it is not so well-known, and the challenges that they face in this odyssey. So hemophilia is a fairly well-known orphan disease, and it's a rare genetic bleeding disorder that is caused by the deficiency of clotting factor A in case of hemophilia A, and hemophilia B, which is also known as the Christmas disease, in case of deficiency of factor 10. Internal bleeding can damage joints and organs, and thus can be life-threatening in these patients. And the most common time primarily affects males, but females who are carriers can also be affected as well. And because it is so well known, the median time to diagnosis is essentially 0 years, with 71% in the first year of life. So when people really hear hemophilia, they feel it's not a rare disease, but it is a rare disease because of the frequency of the population that is affected by it. But it can also be caused by a spontaneous mutations, which would not be expected. So in such cases where there is no family history, females, however, are not diagnosed for 10 or more years, because they're expected genetically to occur mostly in males, and females are expected to be carriers. So there are cases, quite a few of them, that haven't been diagnosed as quickly as the mean time to diagnosis was reported at 4.7 years. Through the survey, the voices that were collected with patients who were with hemophilia, this was some of them that show you the anguish of the patient who goes through that. And one of them said, I visited an emergency room multiple times, but the doctors had no idea that my bleeding could be hemophilia. And this impact of misdiagnosis is very striking in such a case. A woman said, because of the education that females are always carrier, she was labeled as a carrier for a long time, even though her factor level of the clotting factor was in 20s. Another child said that his parents, the pediatrician didn't believe there was anything wrong. He felt we were responsible, the parents were responsible for the severe bruising that you saw in the previous pictures that is seen with patients with hemophilia. So there is a great impact of these misdiagnoses, even though hemophilia is a fairly well-known orphan disease. In the next slide, I'm going to talk about another disease, which is well-known, and there is a lot of awareness that has been created in the US, and it is called as the cystic fibrosis. And cystic fibrosis or cystic fibrosis. So if you go back to your biochem classes, I'm talking to the undergrad students, I remember having that thick Leninger, and I remember that figure where it said about the cystic fibrosis transmembrane conductance regulator. And it is that regulator, and if there is a defect in that protein, it causes cystic fibrosis. Cystic fibrosis is also sometimes called as cystic fibrosis, and there's a story behind that. And the name essentially comes about from a 4-year boy, an observant boy who was listening to his mother talk to clinicians, as well as reaching out to other charity organizations and reaching out to other organizations to raise funds for her child, 3 of her children who were diagnosed with cystic fibrosis. And when he heard her mother talk, he heard her thinking that she was talking about 65 roses. And this is a story of Richard and his brothers. And Richard passed away in 2014. And since that, and essentially since then, before even Richard passed away, it started to be called as 65 roses, a term often used for young children with cystic fibrosis, so that it's easier to pronounce that name. However, how easier that name is, it is still a very life-threatening condition in these children. It's a progressive genetic disease that affects the lungs and other system. There are more than 1,000 mutations in the gene that have been associated with the gene. And most newborns are now screened for cystic fibrosis, which explains the short time to diagnosis of 3.3 years. If we look at the patients, and you talk to those patients, they essentially say that newborn screening, and this goes around a lot in the patient community in the rare diseases, that newborn screening has had a significant impact. And essentially because patients who were born before 2000, where newborn screening was not yet implemented, the average time to diagnosis was 0.75, was approximately 6.6 years, not 7.75 years, but 6.6 years to diagnosis before 2000. However, since the year 2000, the average time to diagnosis was 0.75. So before 2000, when the newborn screening was not implemented, these patients were essentially misdiagnosed with asthma or allergies or bronchitis. So from 2000 to now, it has changed from 47% to 20%. And this gives a lot of hope to a lot of families, because this gene panel that is provided by every state, which can do it for cystic fibrosis, allows them to get better diagnosis. And as you can see in the next slide, about patients who voiced that. One of the women said that I found out while I was pregnant. We were very lucky as we prepared at his birth to we were prepared to deal with these diagnoses. And someone else said that the fact that they now test at birth had just started in 2000, made diagnosis all but immediate. And just that all the states need to be doing this. Just to make it clear that just because it's implemented in one state does not mean it's a federal law in all the states. All the states have their own law, whether they want to implement it or not. So that is a challenge that as a patient advocate that we always advocate for, that newborn screening is essential so as to allow the families to help them diagnose the problem earlier. Another patient said that for us, nothing could have led to a faster diagnosis, and the diagnosis was made as a part of the newborn blood screening that is mandatory in Illinois. He was diagnosed at the age of 12 days and confirmed a day or two later by a sweat test, and we started therapy that week. So what happens is when they start therapy earlier, of course, the quality of life and their chances of living become essentially better as compared to someone who is diagnosed and later. And you will see as I talk later in my talk, the challenges that are associated with rare disease community is the frustration with it of not being diagnosed earlier and running to pillar to post to different clinicians trying to get that diagnosed. The other syndrome that I wanted to talk about is the Ehlers-Danlos syndrome, and it's a group of inherited disorders that affect the connective tissues, primarily the skin, joints, and blood vessel walls. So these patients essentially have overly flexible joints and stretchy, fragile skin, and this can become a problem if you have a wound that requires stitches because the skin often isn't strong enough to hold them. A more severe form of the disorder called vascular Ehlers-Danlos syndrome can cause the walls of your blood vessels or the patient's blood vessels, intestines, or uterus to rupture, resulting in life-threatening complications. Thus, EDS has showed to have the longest time to diagnosis with a mean of over 21 years. Over 77.6% were misdiagnosed, and a mean of 24 tests seen over 15 physicians were among the highest in the study, and over 71% experienced a delay in treatment. So when you hear from the patients in this community of Ehlers-Danlos syndrome, they do say that it's an especially difficult diagnostic odyssey of full misdiagnosis, as I read it in the green bubble, that this patient was often called a clumsy child. When he wouldn't roll ankles or knees or dislocate, when he would roll his ankles or knees or dislocate fingers. As a teenager, accident prone, and that each injury was treated separately. Sprained ankle, torn rotator cuff, dislocated foot, and jammed thumbs. And this essentially, if you read through this patient's statement, you can realize that as a young teenager or as a young child, he was often the mockery of many jokes, and which can be affecting the patient's confidence and their quality of life as well. As you can see that they're in and out of the hospitals continuously with trying to find the right diagnosis and also getting treatments for something that they have not been diagnosed with or still trying to get their right diagnosis. Sufferers of this rare disease stress the importance also of raising its awareness among physicians, as you can read in the blue bubble. They do talk about that we need more awareness by other doctors and physical therapists or PTs. I saw multiple orthopedists for sprains and injuries who never suggested, hey, this could be EDS. So there is an importance of raising awareness for every rare disease that patients encounter. And as the pink one suggests, that the delay in diagnosis has severe impact on the patient's well-being. And I'm sure all of you can understand to some level that as this patient echoes in the pink bubble, that if I had known 28 years ago to not stress my joints, I wouldn't be as bad as I'm today. And if I was known about the common comorbid diseases, I would have gotten treatment for those sooner. So if you see through these patient voices, and the essential reason that I bring this out is to understand that sentiment behind what they're going through, not as a cause of sympathy or pity, but to create a cause of empathy, to understand why it's so essential with the work that Dr. Sakaria has been doing, to the research that he's been doing, why it is so essential to do the research, but at the same time, bringing that awareness, because many diseases are still not diagnosed. Many patients find themselves in a dilemma of getting the right diagnosis. And if you talk to any of the rare disease patients, once they get the diagnosis, their first thing is, oh no, not that I have this disease, but I'm glad that somebody finally found what I have. And that there's the first reaction to when they have their diagnosis is a sigh of relief rather than, oh no, I have this disease. And that just exemplifies what they go through on a day-to-day basis when they're undiagnosed. And giving what I just talked about from a patient's perspective, when you talk about the diagnostic challenges, the patient most often mentioned that physicians weren't listening to the patient. They always found, it again comes back to, because it's the physician's mindset. They weren't looking for the disease because they never saw that before in their treatment with the patients. Bias is common across the globe, not just in where we stay, but across the globe. Patients have felt there was a bias against them due to their gender, due to their race, or their weight or age. Location is important as well. Sometimes a patient found themselves that they were not closer to the appropriate facilities or specialties, and that was sometimes to be blamed for the delay in the diagnosis or the differential diagnosis, which again leads to the second point, the delay or denial on the patient's part also. Sometimes patients just felt like in some diseases, like Fabry, they have intolerance to pain or intolerance to cold or heat. And when they have those conditions, it is just discarded as maybe it's growing pains, maybe you're just having another pain, and they're prescribed some pain medications, or they just take a pain medication thinking that's just another pain, unless it becomes so severe that it starts affecting their quality of life to work or anything. So sometimes the patients do say that it has been a delay or denial on their part as well. Also getting to the correct specialist. For many, it took a long time to get to a physician who was able to make the diagnosis. Like I said, medical education, sometimes even clinicians say that to us, that medical education is important because sometimes they have read that in their residence training about that disease, and teaching physician that is more beyond the 70% that fit into what they've learned in medical school is important. And that is where patient awareness, the rare disease awareness comes into picture as well. Also involving clinicians who have taken, have treated a lot of those patients, getting them into this community and helping them to, educate others as well. The state of medical knowledge, in some cases, again, the blame was placed on the deficient knowledge of the disease at the time. And this is not to blame on the patients or the physicians. When I talk to either of them, on both the sides, there's only one common theme: the awareness that is needed for every rare disease state. So there could be better diagnostic tools that can be available, better quick time to diagnosis so that that delay that is seen is not there. And of course, in the US, some believe that insurance also contributes to it, because sometimes you need the insurance to approve certain tests because the person has certain diagnosis. So some do believe that insurance, or the lack of it, contributed to the lengthy diagnostic path. From a physician's perspective, there has been, I know, a mixed debate, a mixed bag in the previous session, but essentially the physicians do think that there is a lack of proper genetic testing. And genetic testing can have a wide range of variants, ranging from newborn screening to whole genome sequencing to 23andMe testing, which is a genetic test that allows them to predict, genetic testing that allows them to select relevant variants in the genomic DNA of adults from saliva for the sole purpose of reporting and interpreting genetic health results, health risks, and reporting carrier status. So it is not intended to diagnose any disease. And this, I'm talking about the 23andMe testing that they do. Finding the right genetic counselor is also important, is something that the physicians have often said, because finding a certified genetic counselor who can help walk a patient through the best diagnostic test for his or her individual case is of utmost importance. Sometimes your primary care provider or the family doctor that you see may not know the right physician or the right specialist, or recognize the disease to put them in front of the right specialist. So the way it works here is that you have a primary care provider whom you go to when you first find any symptom, and then he directs it as needed to the right specialist. So it's not like I can call the cardiologist or someone else, but I have to go through my primary care provider or my family doctors or my pediatrician in a child's case before that I can go into another specialist and go up to him. The lack of timely diagnosis, which I alluded to it, that the importance of timely diagnosis is critical, as they can be pivotal in preventing further damage. For most diseases, especially the rare disease, preventing damage is much easier than repairing it. And for most diseases, especially the rare ones, like I said, sometimes the damage has been done by the time they're diagnosed that it's too late to come back. So newborn screening essentially most times helps, because then they can start therapy earlier to prevent that or later damage to them. And we've talked about this, the shortage of healthcare providers and physicians in rare disease, and the knowledge gap that exists in the community with rare diseases. So what are the solutions? And some of that comes off often from physicians and patients is from patient's perspective, that when you see your physician, make suggestions. When you see your primary care physician, tell them what you're going through. Know your body, be your advocate. And that is a patient's perspective, because sometimes being your own advocate has resulted in educating the providers. And I can tell you of a story that I worked with two of my colleagues who work in rare disease. And one of them had a child with a very rare disease, and he had what is called as the Sanfilippo syndrome. And at that time, when the son was diagnosed, they didn't know much about it. And I remember my colleague telling me very distinctly that when they were in and out of the hospital, he and his wife often found themselves, educating. And this was a young couple with no scientific background, trained in business. But when their son was diagnosed, they become the experts in that rare disease. They found themselves at rounds where they were educating physicians, medical residents, fellows, with the diagnosis their son had, and explaining to them over the years what they had experienced and how that has led to it. And this became a constant feature. Another colleague of mine whose daughter had an immunodeficiency syndrome essentially found the stem cells to be the cure for them. And that came from her own sister for the brother. So it was a very beautiful story where the sister's stem cells could provide a new life and could be a cure for his own brother. But of course, that was, as beautiful as it sounds, it was a lot of torment on the family before the diagnosis. They were from Mexico, moved to Boston for getting the right care, and they were able to do it. And then now he goes on to Boston Medical, Harvard Medical School, Boston Hospital, MGH, Massachusetts General Hospital, educating clinicians, fellows, and residents. So sometimes it is necessary to educate, and most often that is a theme that is seen in the patient community, that tell your doctor about it. And most good physicians are willing to listen and educate themselves, or reach out to their peers or colleagues to help them understand this disease more. So it is a very collaborative environment, as I will talk about a little bit more. Medical education, like I said, it is important to raise that awareness among the physicians for the general public, to help you put your disease on that map, and making sure physicians and patients consider it more than the cause of their symptoms. Patient advocacy organizations play a very critical role, especially because these patient advocacy organizations is not only bringing awareness to the disease syndrome, but they also provide the particular rare disease community, a particular rare disease, the support that is needed. So oftentimes there are 4 or 5 patients in a particular country or an institution that have that rare disease, and you find yourself lonely. Because that is not the norm of life. You don't see the other kids or other patients or other elders going through that. So they do find that in a limbo where they can't relate to it. So that's when patient advocacy organizations not only provide support, we as humans rely a lot on relativity. When we can relate to someone, we identify with them, and there's a greater connection. And the patient advocacy has that attribute to it. They not only can connect you with the right support system, but you also find that there's another person who's going through what you're going through, and you're not alone. And then they collaborate and bring about changes that are not essentially happens what a physician or a patient individually can do. So there is this collaborative environment with patient advocacy organization that I've been privileged to be a part of, and I've had a privilege to collaborate with, where we can go and voice our opinions and activate for research funding like never before. And because the voice of a patient is very important in making a difference in the rare disease community. Again, from a provider's perspective, in a physician's perspective, matching a patient with a diagnostician who can diagnose. There are several diagnostic tools, but what is the right tool for that particular rare disease? What is the right tool for that particular condition? Because there could be result in a lot of false positive. So you need to know the right diagnostic tool as well. And the right healthcare provider, connecting with healthcare professionals who've seen these types of patients, which I talked a little bit before, in their practice helps. So reaching out to that person who can, educate you more and willing to tell you more about that. To reaching out to that peer in your medical community is something that physicians have often alluded to. And it always comes off better when it's from a peer than from someone else. So training, or training also, like I said, training young doctors or young residents, showing providers to recognize when something doesn't look right. Providers also often say that using and sharing phenotypes built through a worldwide database also helps, because then you have an access to this and then you can identify patients more quicker. So AI has been in the talks at several conferences that I've attended to identify using that, and it's still in the works. It's very new to identify those phenotypes based on what has been collected. Again, collaboration is very big, is essential for this community between the patient advocacy organizations, physicians, researchers like Dr. Sakaria, academicians, and patients, so that the data that is collected, the patient-reported outcomes that are collected, can be helpful in 10 years from now, diagnosing the new patient population that is emerging and lead them to quicker diagnosis, which has happened with some of the diseases, which has led to quicker diagnosis. So that collaboration is an important part of it. So what is the impact of rare disease on caregivers and providers? And that is also something that is often neglected, and which is brought to light through a study that was done and often brought to light by patient advocacy organizations and clinicians who are very involved with these patients, that there is an impact on patients and caregiver at different facets of their life, which I've listed out here. There's an impact on the family. When you have if some of you have or some of you haven't, when you care for a loved one who is sickness, you know that it takes a lot of your time emotionally, physically, and at that time you might not be realizing it. But when patients are asked to reflect back on it, they do say that they're unable to pursue their hobbies or favorite activities due to the caring of a loved one. Some mentioned that it has brought their family together, or some mentioned that they had marriage issues due to rare disease, and the time required to deal with rare disease of their loved one takes time away from spending it on personal activity. So imagine with these patients, you're going in and out of the hospital, treating them, getting them the right treatment, trying to find them, trying to address that pain that they've been describing, but haven't yet identified the cure for it, or living with that glimmer. COVID-19 is the perfect example. We all lived in a time where we were all shut down, and we did not know when the hope would come out, when the vaccine would come out. People were tired. And that is what the rare disease community goes through every day. Because for them right now, there is no cure. Some of those diseases do not have a cure. And they live at that brink of hope that one day there will be something that will be discovered. Until that time, you can probably relate to some level the frustration, the anguish of these patients and these families that they go through on a rare disease community, and the hope and the glimmer that came in when vaccines were, told to the countries that the vaccines are available. So that is pretty much what a hope that they live in a situation like that. When it's a constant phase of anxiety, there is emotional distress, and that is the emotional impact of rare diseases. One of the panels that we organized through our collaboration with other patient advocacy organizations is post-traumatic stress disorder, because not only does the patient go through what they're going through, but the care provider also has a significant impact. It has a significant impact on it. And it's the most common topic and the most attended topic on our panel discussions is the post-traumatic stress disorder, because the patient goes through a lot of mental stress on the care of on the caregiver is intense. The mental stress on the caregiver is intense. And like I said, a lot of the loved ones feel more isolated from friends and family. Many parents, and I've seen this across the board, speaking to various families over the years, that many parents do seek help professionally while providing care for their child, or in case of an adult, or caring for their loved one. So it does impact the patients on an emotional level, and there's of course an impact on the finances. It's a very high impact, because when dealing with these diseases, a lot of time, because of the care that the caregivers have to provide for the loved ones, they've let go of opportunities of promotion or employment, or they have to let go of education because they have to care for their loved one. And those also affect emotionally on the caregivers and providers. Many of them opt to make work part-time instead of full-time. I've had the privilege of seeing patients in the hospital where patients cannot afford to miss work, so they leave their loved one at care for with the physicians. So there are parents who have to leave their little child or baby and go for hours while they're not at their bedside taking care of them, because they have to provide for. So it has a deep impact on their finances and their employment. And educational impact, patients and providers become frustrated over the lack of knowledge of awareness about the rare diseases of many professionals in the healthcare system. And again, if you've seen, I've emphasized the importance of the rare disease awareness is so important, because when we create this awareness, there's an importance for research funding, and the importance for research funding helps to get the cures that are needed, the therapies that are needed. And that is why it is so important to create that awareness in any media that is available. Again, just giving you, going back to patient voices, the impact of these rare diseases from the horse's mouth. Someone has said that my disease has changed my whole family's normal. For what is their new normal? Sometimes it's just going back and forth from hospital to hospital, trying to find the right diagnosis, trying to get the help for their other child who's not suffering, or the next child who's coming in. There are anguishes that I've spoken to patients who've said that we're not sure of another child because of our one child. But there are also patients who've said, as much as my younger one went through physically, my older one suffered emotionally because I wasn't able to care for him. Again, rare disease patients often say that my entire family had to take a crash course in understanding everything about the disease. And the most difficult part for my parents was coming to terms with there being no cure for the disease. Another patient says that my family has had to take me to several doctors to find the diagnosis and get where we needed to be, and they had missed work without getting paid. I was unable to get hired because of my condition, and was unemployed and did not qualify for disability until after 12 months of being disabled. We've had to have a fundraiser to help raise money to get the medical places I needed to be in several different states. And one person puts it that this disease steals your freedom. And I have personally been a rare disease advocate for my own reasons, because I have a very close association with Noonan syndrome. Noonan syndrome was identified in 1 in 2,500 live births. It's an autosomal dominant mutation, and that means it has a gain of function mutation in several genes. And PTPN11, KRAS, NRAS are most commonly found, but BRAF is found in less than 2% of the world population. So there are very few, and it's a spectrum. So you could have patients who have mild symptoms, and there are patients who have very severe symptoms. But either ways, there's 3 times more mortality than general population. It's a rare congenital disorder affecting the whole body. And my association with rare disease stems for our own foundation, which is called Conquering with Canaan. And this was out of my own personal experience, being a rare disease care provider to my own son, Canaan Joseph. And he passed away in January of 2018. And my husband and I started this foundation in his honor to support other rare disease families, because we understood we were very privileged to get Canaan's diagnosis, be placed in a place where we were in the country with the top hospital, Cincinnati Children's, and get his diagnosis at 8 weeks. But having been in the hospital with my son for 8 months of a time, I and my husband and I understood the vagaries that patient family goes through. We were privileged to have children in our backyard, literally children's hospital in our backyard. But I know of patient families who have traveled across the globe to get treatment at Cincinnati Children's, and since then we have not only provided patient support. This is my older son to Cincinnati Children's Hospital in terms of care packages, in terms of food, because these are patients who've been staying in the hospital for a really long time. And as much as I'm a researcher who did research in my own son's condition of hypertrophic cardiomyopathy, which was one of the cardiac manifestations. My son had such a rare condition, which was due to a spontaneous mutation. So it was one of those cases which was not identified in a family history, but he was diagnosed with it. And when Kaden was diagnosed, standing at his bedside, when he breathed his last, I realized the gap between the bench science and the bedside. And that gap is what motivates me every year to go to the Congress at Capitol Hill in Washington, D.C. and advocate with congressmen and tell them the importance of research. Because as much as I was doing research with a very well-known scientist in Heart Institute, my son's care did not provide the therapy or cure it needed. So for me, it has become, and my family's motto has become, to advocate for these rare disease patients, advocate for the rare disease community as a whole, and support them in every way that we can. And with that, I would give it over to Preeti and would take any questions or comments. And nothing is off limits, so you can ask me any questions or comments that you want to about my son or my family or anything that I've spoken about. So over to you, Preeti. Thank you. And I also want to recognize my parents and my in-laws who joined me from India as well.
Yeah, thank you so much, Swati. It was really such a good and enlightening lecture, and also it was a very emotional journey for us also. And I'm glad that your parents and your in-laws are also there and they've also seen me here. So I'm glad they have joined here on Holy Evening, and they are the kind of seeing the kind of work that you're doing, the kind of noble job that you're doing along with your profession. It is not just a professional thing for you, it is more than that. I understand that. So I would now like to invite Vinita to give a vote of thanks to you. Yeah, Vinita. Vinita, are you there? There is some connectivity issue. Probably you can take the questions before that. If there are any questions in the chat box.
Sure, ma'am. Ma'am, there is one question. First question is, can you throw more light on 23andMe testing? Am I audible, ma'am? Preeti, ma'am?
Yes.
Yes.
That's a great question. To be honest with you, I don't do the clinical testing, so I am just, but the 23andMe testing is more not a diagnostic testing, not to diagnose the patients, but it's essentially a test that is done in every state. If the parents want, if you identify in your family that there is a particular gene, what is the risk for another family member to have that disease? And I can send you the paper of the article that first published this, but essentially it is to more to categorize the risk analysis for a carrier or a carrier in the family than someone with it's not for diagnosis. So I don't do it, so I don't know the essential what I can say is the way it is done, but mostly to say that it's more for identify the risk factors and how it would affect the future families, and that is what it is. That's why it's called 23andMe testing.
So what is it exactly? It is 23andMe testing? What do you pronounce it?
23andMe.
23andMe.
Chromosome.
Okay.
The chromosome 23andMe.
Okay.
So it's like, what if there is another? That's how it's started.
Okay.
Okay, ma'am. Next question is, what is the treatment for cystic fibrosis? Anyone being cured? What is the life expectancy?
That's a great question to ask a clinician. I am not a clinician that treats cystic fibrosis, so I can't tell, but the life expectancy is much better than what it was before, just talking to cystic fibrosis patients, but there's no cure yet for it.
Okay. Coming back to 23 and me, there is one question. How much do we trust ethical and privacy processes when we turning our most confidential information, the DNA to private companies such as 23 and me, MyHeritage, etc. DNA information can be accessed and can be used to discriminate in healthcare, insurance policies, hospital checks, etc. Is this a common issue being discussed when advocating rare diseases and commonly available genetic testing kits?
That's a great question. And essentially, the United States has a HIPAA policy that is protects the patient's identity. So when these tests come or when they go, they are de-identified, and then these tests are done. So nobody knows when you submit it except your clinician to it, who it goes to, and that to only the referring physician. Even your family doctor would not know except for the diagnosis. So in the US, the HIPAA privacy policy is, so even when we get data sets, it's de-identified. So we do, the HIPAA privacy policy does protect you from it. So yeah.
Okay. Next question is, ma'am, what can be physical mindset of physicians for treatment of rare diseases and also at the patient side also?
I'm not really understanding the physical
mindset. I think this is related to like one person who is once experienced with the rare disease in his or her family, then only they become they have that kind of mindset. So something they want to ask like that, what can be physical mindset of physicians for treatment of rare diseases and patients also?
Okay, so I think from the patient's from a physician's perspective, if a physician is treating rare diseases, they're always willing to advocate for them. They are our biggest, biggest cheerleaders when they come into it. And when they come with us to the Congress or Capitol Hill, there's a greater impact because they are the experts in the disease state, essentially, and they're willing to educate the community. So I have never seen a physician say no if he's treating a rare disease patient, not wanting to come and educate the community. In the several panel discussions that we hold as an organization or collaboration with other organizations, they're always willing to come, but they do, and that is where we understand, because they are treating the patients, we understand what is coming from that. From a patient side who has gone through it, they are also willing to, someone who has had a rare disease, they're willing to share their experience, but also you have to understand there's a time frame for them too. Not every patient is willing to share at the time when they're in the crisis. Some are, and everybody's different, and some aren't. So patients are also willing to share it. It is a very stressful condition that they go through, but essentially, once they are over that bump, that I may say, in that early days of life, and once they're trying to get their new normal set in, patients are willing to share their experience and become advocates themselves. And we see that earlier in March, when we advocated for congenital heart disease at the Capitol, they were just from my state, there were 30, and overall in the US, they were around more than 200, I believe, who came and advocated. So, and this is including policymakers, providers, and patients. So people are willing because that gives them they give others the hope, and also puts in that impetus for the need for research funding, as well as the necessity for cure.
Okay. One more question I have from my side, that you have a foundation concurring with Canon. So shed more light that how it functions, in what area you are doing this kind of work. So can you elaborate on that point, that what is your foundation about, how it functions?
So essentially, like I said, it was started in honor of my youngest son, Kanan Joseph, and my husband and I started because we saw firsthand what families go through while living in the hospital, with our own son who had a heart condition, which was the reason for his death. So what we did was, initially we were supporting care packages. There were patients who stayed in the hospital for 4 to 6 months, haven't gone back home. And we've had stayed in the hospital ourselves for 4.5 months. And we provided care packages, which meant they have young siblings and they have younger children or other kids in the house. So what can we do? We provided them things that they didn't have to run out of the hospital for, where they could be at their bedside. Sometimes the normal for them is that all they can do is read to their children because the child is hooked up to so many machines or so many things. So things like that, which could help them in their daily life, thinking of meal cards or restaurant cards, or providing, when you stay in the hospital for so long, you can get as much food as you want, but after a point you get tired. So we used to get them for Mother's Day, for Father's Day, special meals for the patient families. Also, the care providers, the healthcare providers, they go over and beyond trying to support the patients and their families. So we try to provide them as well as meals, appreciating all the hard work that they do. So, and then we also organize, apart from fundraisers that we do, we try to get panel discussions with experts in the field so that there is an education in the community for it, which is necessary. And this panel is not just providers, but also patients, as well as patient, parents of the patients, people who've been in the disease and can give their insight. So throughout the life cycle, people who've lost loved ones, people who have had a child or had a spouse or had a loved one who had a condition, or an expert in the field like a rare disease clinician or a diagnostic genetic counselor, we get them on.
Okay, thank you, ma'am. There are no more questions. I would like to give mic to Preeti, ma'am, so she can take lead.
Thank you, Prabha. Swati, I also have one more question. Are there any people who have formal degree in law and they are working in rare diseases? I mean, there are actually some patient advocates.
Yes.
I mean, you're also involved in patient advocacy program, but you may not have a law degree. So do you actually require such things?
You don't. A patient advocate is someone who can just advocate, basically cheer for you or advocate for that patient. So that's what a patient advocate. So anybody essentially, a lot of our patient advocates are not even science degrees. They are basic high school degrees, but because of their experience with their child or what they've gone through, they become the experts. So we always say this in the rare disease community, they are the experts because of their experience with their children. And it's not the disease that would define them. The disease is just a part of them. There's more to them than just the disease. So when you look at someone with a disease, always remember to look at them more than what they are. It's not the disease that is defining. So anybody can be a rare disease, someone who's very passionate, who's had the experience, and yeah.
Yeah, thank you so much. I would now like to invite Anas to give a formal vote of thanks. Yeah.
Am I audible?
Yes, you are.
So good evening, everyone. On the behalf of this Department of Biochemistry at Deshbandhu College, I would like to extend a hearty vote of thanks to the two speakers for joining us today and providing us crucial insights on the topic of rare diseases, the associated research challenges and concerns. I would like to start by thanking Dr. Vinod Skaria for an enlightening talk on personal genomes to population and experience from India. Sir, it was a true delight to hear you today. And sir, thank you for sharing with us with those crucial examples about from India, and also throwing light on the topic that why it is imperative, what opportunities does India provides us to study genetics because of the close-knit family structures and because of largely undisplaced people, and why it is imperative for us to create an ecosystem for clinical genomics in India. And sir, I must mention that your efforts in the process are certainly commendable. I would like to further express our gratitude, a deep sense of appreciation for Dr. Swati Kundari for apprising us about rare diseases, for sharing those patient voices, which tells us a lot about social and emotional needs of people suffering from those rare diseases, and also the impact of those diseases on caregivers, family, finances, etc. So, clearly, these examples tell us about our duty as future researchers and what shortcomings we have to address in the system that exists as of now. Ma'am, thank you for inspiring us with the amazing work you are doing for the community. And thank you for joining us today and making our annual event so rewarding. Further, I would like to express our gratitude to our principal, Dr. Rajiv Agarwal, who has been a constant source of encouragement for all of us. So we are all very thankful for your support and commitment to students, and also jubilant that you gave your valuable time to us. I would like to extend my deep gratitude to Dr. Indrakant K. Singh, Coordinator DBT Star College Scheme, and Dr. Aditya Saxena, Coordinator IQAC, for joining us today. The role of our amazing faculty is also worth a mention here. Our teachers are our mentors, guides, and have played an indispensable role in making this event a great success. A big thank you to our convener, Dr. Preeti Karwal, our co-convener, Dr. Vinita Kashyap, our organizing secretary, Dr. Vanshika Lam, and all members of the organizing committee, Dr. Kamna Singh, Dr. Rajni Jain, Dr. Prabha Aryan, and Dr. Asta. Last but not the least, I would like to thank all of you, audience and participants, for joining us today despite your engagements in this festival time and making Biospark 2021 a great success. Thank you, everyone.
Thanks a lot, Anas. And it's the selfie time. We need a screenshot for it. Please, all of you, please switch on your videos. I would request all the participants and the speakers to please switch on their videos. Vanshika, can you please take care of it?
Yes, ma'am. Ready? 1, 2, 3. Just a second. I want to take one more. One, 2, 3. Thank you.
Thank you so much, everyone. Wish all of you a very happy and safe Holi. And thank you, speakers, once again, Dr. Vinod and Dr. Swati, for joining us.
Thank you.
Thank you, ma'am.
Thank you.
Thank you, ma'am.